Nice introduction. So today here with me is Joy Omani, and we together will present a collaborative cure project that we are developing for this fall semester. So this is, we will present a kind of model of a collaboration between teaching lab and Joe and the MCC community, and also a research lab in our department. It feels like these different pieces coming together. So it's we are excited about that. Yeah, I will let Joy briefly introduce herself.
Okay, like Joe said, I'm Joy Omini, and right now I'm a 5th year PhD student in Obata Lab, Biochemistry Department, UNL. My research is based on the malate dehydrogenase citrate synthase metabolon and its role in metabolic flux regulation.
Okay, I will first briefly introduce the cures that we are doing here at Nebraska. So since 2016, with the support from the MCC, we integrate a 6-week modular cure. So this is a mini cure that we started with. So the structure of this cure is, for the first part of the semester, students will learn the basic lab skills like the pipetting, pH meter, and they will learn the DNA protein techniques. Then the latter half of the semester, we will start to introduce the MDH project. So we started with the watermelon glyoxysomal MDH project that's focused on the active site, active loop site. So we see here, yeah, there are several things that we do in the teaching lab. Students will read the primary literature, look at bioinformatics, and present a hypothesis. Then they will measure the kinetics to test their hypothesis. And at the end of the semester, they will present their project. So what we are planning to do right now, it is to expand the 6-week modular cure to a full course cure. From the first stage of the IUS grant, that we get some very interesting results, and one of them, it is the longer time of the cure that students exposed to in general gives out a better student outcomes. So this is one of the driving force that we want to expand the cure. You can see here, you know, the green, that is the MDH component. It's almost expand to the entire semester. So also what the change we're going to do is we want to switch, it's a very interesting connection that between Joe and the Joyce research lab, that's from Dr. Obada's lab, and teaching lab that we just got all connected last year. And so now, next, I will let Joy to introduce the background on the Metabolon research that she's doing in her lab.
Okay, thank you so much, Jing. Yeah, I would just like to make a brief introduction on metabolons. But first, I have this slide showing the cellular metabolic network. I'm sure we can see a lot of pathways that we are familiar with. There's the pentose phosphate pathway, the fatty acid synthesis pathway. We see the beta oxidation pathway, and also the citric acid cycle pathway. But what is not very obvious in this cellular metabolic network is the presence of metabolons. And it's interesting that metabolons are present in many of these pathways. You know, any pathway you can think of, metabolons are strategically positioned right there. So we have the glycolytic pathway here, it has a metabolon. We have the citric acid cycle, it has a metabolon. The fatty acid synthesis pathway also has a metabolon. But then this begs the question, what are metabolons and why are they ubiquitous in the cell? Also, what is the role that they play? So to briefly introduce metabolons, I have this slide here showing 2 enzymes, enzyme A and enzyme B. But before I go into that, I would briefly define what a metabolon is. A metabolon is a transient multi-enzyme complex that's comprised of 2 or more enzymes that catalyze sequential reactions. A key note there is transient, meaning that metabolons can associate or dissociate depending on the needs of the cell. So here I show two enzymes, enzyme A and enzyme B, that catalyze sequential reactions independently. So these two enzymes do not form a metabolon in this case. We have substrates here that are called substrate S, present in the bulk phase of the cell. So let's assume that the background of this slide is the bulk phase of the cell. So the substrate is able to diffuse into the active site of its enzyme, enzyme A, where it's converted into intermediate I. Intermediate I then diffuses out of the active site of enzyme A and into the bulk phase of the cell, and then it accumulates. Intermediate I can then diffuse into the active site of the next enzyme, enzyme B, where it is converted to the product P. Product P then diffuses out to the active site of the cell, to the bulk phase of the cell. In the next scenario, I show a metabolon here where enzyme A and enzyme B form a multi-enzyme complex. So these two enzymes interact and they also form a metabolite channel. So one thing to take note of here is that metabolons always have metabolite channels, and then they are able to channel intermediates from the active site of the first enzyme to the active site of the next enzyme. So again, I'll go through the process. We have substrate S here in the bulk phase of the cell. The substrate can diffuse into the active site of the first enzyme, enzyme A, where it's converted to intermediate I. Intermediate I can then be channeled from the active site of enzyme A into the active site of enzyme B, where it's converted to a product P. Then the product can diffuse out of the active site of enzyme B and into the bulk phase of the cell. This shows us one of the main advantages of metabolons, which is that it can protect intermediates from competing pathways. So if we look closely, we'll see that intermediate I didn't diffuse into the bulk phase of the cell, unlike in the previous scenario. And so if we have an enzyme, let's call it enzyme yellow, that also uses intermediate I as a substrate, because intermediate I is channeled within the metabolon, the activity of enzyme yellow is downregulated. So this is a very interesting advantage of metabolons. And we know these advantages that have been postulated theoretically, but we do not know the function of metabolons. We don't know yet what the importance of metabolons are in the grand scheme of things in the cell. So now, what's the function of a metabolon? Like I said, we don't know yet. Right now, my lab is studying the function of metabolons in metabolic flux regulation, and we are using yeast model. We have some interesting results that have been published, but we are still in the process of studying the role of metabolons in metabolic flux regulation. And like I said, we are using the yeast model, which is a very interesting model. Some people may ask why we decided to use the yeast model, and I'll go into that in a bit. Apart from the fact that the yeast cells are easily manipulatable genetically, they are also easily manipulatable metabolically. So the yeast cell has a very robust metabolic system. Depending on the carbon source that's available to it, it can modify its metabolic network and use different metabolic pathways. So that made us choose yeast cells, because then we can change metabolic flux and see how that affects our metabolon. Also, the yeast cells is a mammalian cell, like we all know, and it's very homologous to its genetic information to humans. So we hope that studying yeast cells will allow us to extrapolate whatever results we get to apply them in humans. And using the yeast model, we're going to be studying the TCA cycle metabolon, which comprised of the malate dehydrogenase and citrate synthase enzymes. These two enzymes catalyze the last and the first reactions of the TCA cycle. And as you can see here, they are strategically positioned at the key regulatory points of the TCA cycle pathway, which points to the fact that this metabolon might actually have a metabolic regulatory role to play in the cell. So to zoom in on the metabolon that we are working with, I have this slide just to emphasize. Like I said, we're working with the malate dehydrogenase citrate synthase multi-enzyme complex, or metabolon. This metabolon forms an electropositive channel, that's this channel, and it's able to channel oxaloacetate, which is the intermediate, through the electropositive channel. So we use this metabolon to study the function of metabolons in general in metabolic flux regulation.
So picking up from what Joy has been doing, she actually published a nice paper, 2021. And in her paper, there's a simulation of the interaction of porcine MDH and porcine citric synthase. And they proposed some interactions on the amino acid residuals. So for example, here, arginine 67, 65, these are the two key residues on the citric synthase site. Now, the proposed interaction sites are the aspartate 88 mainly dehydrogenase. This has not been tested yet. So based on this information, we did a sequence alignment between the three species: yeast mitochondria, porcine mitochondria, human mitochondria, MDH, and we mapped the proposed interaction residues to this sequence. So two of the most interesting ones to us, one is what I just mentioned, aspartate 88, and it's happened to locate it on the active loop site, right? And the other one is toward the C terminal, it's the glutamate 289. So these two are conserved across all these three species. So what we plan to do for this fall is to make mutations and use it for the cure. The collaboration with Joe. So Joe has been looking at phospholipid, and he probably will talk more about it in his talk. And some of these examples, the sites, they happen to locate it in regions near to these interaction sites. So what we will do in the teaching lab for the students, they will look at the visualization of the MDH structure, and they will also test the enzyme kinetics for these mutants.
Okay, and then when the enzymes are generated by the cure lab that's in the class, we would use the enzymes to test interaction and then the effect of the mutation on the MDHCS metabolon. And we'll do that using this biophysical technique called microscale thermophoresis. Basically, what happens with this technique is that a fluorescently tagged molecule, any biomolecule at all, can be used, but in this case, we'll be using malate dehydrogenase or citrate synthase. So when it's fluorescently tagged, thermal gradient is applied to it, and then the equipment measures the migration of the fluorescently tagged molecule across the thermal gradient. So the principle that it works with is that the bound molecules would migrate slower across the thermal gradient, while the unbound molecules would migrate faster. And so with that, it's able to generate a KD curve, dissociation curve, and then we get like a KD of interaction, and we can measure metabolon association and dissociation. Right now, we have these data showing the effect of substrates on the interaction of our metabolon. We did this with the porcine hearts, malate dehydrogenase, and citrate synthase. And we see here that the presence of substrates increased the interaction of our metabolon. So the green graph here shows the control, and then the other colors show the effect of the substrates. And we see that the curve shifts to the left, which is an indication of an increase in interaction. We also see something interesting here where the presence of products shifts the curve to the right, which means that it decreases the interaction of our metabolon, right? And this would probably bring something to your mind where we know that substrates increase the rate of reactions and products reduce the rate of reactions. We see the same trend here. The substrates increase the interaction of our metabolon, while the products reduce the interaction. So that also points to the fact that metabolons might be a regulatory machinery in metabolic flux regulation. And we hope to use the mutants created by the cure class to test the effect of the mutation on the function of our metabolon using the yeast enzymes in this case.
Okay, this is the last slide. So this is the model that we put together. 3 partners in here, teaching lab, MCC, and Obada lab. We collaboratively working on different aspects of this project. So Joe and MCC has been providing materials, protocols, and Joe will also making phospholipid mix in yeast. and we will test it. And research lab, they will actually, they have collaboration with another group. They will do the simulation on the protein-protein interactions, and they will also carry out these binding assays. And the link between research and teaching lab, it's actually a very important GTAs. They participate both teaching and research, and also this provide them a unique professional development opportunity. So teaching lab, yeah, we do cure, and we also make some mutations, and also we carry out some kinetic measurement. Thank you.